All right, so it's being recorded now, and I should be sharing the screen. So let's get started. My name is Lenny Teytelman. I know those of you who signed up, it originally said that our head of outreach, Dr. Gabriel Gasque, would be doing the webinar, but he's actually conducting a workshop in Mexico on method writing right now. So you're stuck with me. I'm the CEO and co-founder of protocols.io. I'm a geneticist, both computational experimental biologist by training in my PhD. And I co-founded protocols.io 10 years ago and went live in 2014. So this is introduction. Welcome if you're just learning about protocols.io. And just housekeeping before we get started. As I mentioned, we are recording it and we'll send the link to the slides and the video to everyone who registered. So you don't need to scribble down URLs as we go through it. There will be a Q&A at the end, but you don't have to wait till then. You can add questions using the Zoom panel. There's the Q&A button on the webinar, and you can ask questions as we go along, and then we'll get to them either during the presentation or at the very end. If you're already familiar with protocols.io, or you're interested specifically in how to share publicly and what options exist for peer reviewing protocols, we won't get to everything on the intro webinar, but we have advanced and other standing and special webinars. So check out protocols.io/webinars. The link is here. And you'll be able to see what are the different ones that we're offering every month. And finally, if you have, as you start using protocols.io, of course, you can always email info@protocols.io with any questions, but at this link, the /help/demo, there is an easy way for you to sign up for a one-on-one 30-minute call as needed if you want to ask specifically something about the editor or any functionality and aspects of protocols.io. You don't have to wait for the webinars that are regularly scheduled. You can just, at a time of your convenience, on a date that's good for you, jump on a call with someone from our team. All right. And on this webinar, we'll start quickly with a general introduction around the challenges of method sharing, both internally and externally, as we publish, as we do research, and as we organize the protocols before we publish. If you're academic, if you're in industry, you might not be publishing at all. And then we'll switch from slides to a live demo, and we'll go over the key functionality on protocols.io, touching on some of these things towards the end, and then the Q&A. All right. So again, you should be seeing my screen, and if you have questions, use the Q&A button on the Zoom panel to add them. Great. All right. So at this point, we can get started. And as you can see here, I have a long collection of people on Twitter complaining about method sections in published papers. So here is a postdoc from UC Riverside who says, I'm looking for a protocol in a 97 paper as described in 96, finds 96 paper as described in 87, finds 87 paper, and even University of California doesn't have access to that. So it's a common and very frustrating experience. This is from a biologist. On the right-hand side, you can see a physicist with a very similar complaint. Devices were fabricated as previously described. Now, the original reference in the 2009 paper is devices were fabricated with conventional methods. So good luck trying to replicate this. Obviously, this is an issue with the way we publish the method sections of our research papers. But the challenges of finding the protocols are not simply a function of published papers. So in 2015, an initiative was started between Center for Open Science and Science Exchange called Cancer Biology Reproducibility Project, which tried to independently replicate experiments in 50 cancer biology papers published in top journals. And it was a multi-million-dollar effort to recapitulate and see what reproduces and what doesn't in the reproducibility initiative. And writing about it when the first results started coming out in 2017, a year or two after the project launched, Ed Young covering it in The Atlantic, there was a lot of attention to this effort. And in The Atlantic, Ed Young was writing about it saying, excerpt from his article is, the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes are not described at all. Researchers simply cite an earlier version of the similar technique. And this, this is similar to what we saw in the tweets on the previous slide. And of course, it is really hard to build on previously published work. But one of the really interesting things about this whole initiative is that it wasn't just the published paper that was challenging. So, nope, I didn't mean to go here. Sorry. So one of the things that stood out about the entire effort is that the people running the reproducibility project actually contacted the individual labs that published the papers. And even those labs had trouble finding the precise protocol that was used and reflects the method in the described in the published paper. So on the summary page from Center for Open Science on the right-hand side, you can see that from the 50 published papers, 0% had completely described protocols. They had to contact the individual research teams. And frequently, even when they got in touch with the research team, the team could not figure out, the person has left who did the work, or even if they're still there, they don't remember which version in their lab notebook corresponds to the published paper. And so the initiative wound down at just 18 out of the 50 papers that they set out to replicate. And what it highlights to us is that organizing protocols is not just around how we publish them, that's external reproducibility, but there are also much earlier challenges to keeping track of precisely what we've done and organizing it and collaborating on method development. And that we call the internal reproducibility. And so the mission of protocols.io is simply to make it easy to share the method details before, during, and after publication. And just as a 10-second aside, the reason I co-founded protocols.io is that in my postdoc, I spent literally a year and a half correcting just one step of a previously published method. And so instead of a microliter, needed 5, instead of 15-minute incubation with that enzyme, needed an hour. And a year and a half into it, I have this correction, but it's not a new paper, it's not a new method. It's correction of something previously published. And so I don't get credit for it, and everyone else who's using the same technique is getting misleading results and has to spend a year or two rediscovering that exact tweak. And so that's sort of what led to my obsession with creating a central place for exchanging this. But as you can see in our mission statement, it's about organizing and sharing our protocols before publishing, making it easier to detail precisely what we've done as we publish, and then having a place to come back to, because methods are living, breathing creatures. having a place to come back to share your corrections and optimizations, no matter how long after you published it. So it's a snapshot. This is where protocols.io is today. There are two sides to protocols.io. There is the open access, free to read and free to publish side, if you're sharing protocols publicly and contributing to the public knowledge base and repository. And there are just over 14,000 since we started in 2014. There are over 14,000 publicly shared protocols at this point. And most people, obviously all protocols start out privately. We'll go over that in the live demo in a second. There are over 55,000 private protocols. And it's the private side. You get a couple of private protocols when you get started with a free account. It's the private side that we charge for. Many of you on this webinar are at universities or companies that have a site license. So you have unlimited use of protocols.io. If you're on a free account, you have unlimited use of protocols.io for public sharing, but hopefully your organization signs up, or you can pay directly for a premium group. And the public methods, I should say, the public methods on protocols.io, there's over 1 million researchers every year that come to, they don't need to sign in, they don't need to register, they're coming to view the public protocols that people like you share. And the last slide before I switch to the live demo, just where we are today, there are many, many biomedical journals that recommend and encourage in author guidelines and submission guidelines when you submit your manuscript, that recommend and encourage using protocols.io to make your method sections more reproducible and easier for someone to build on. There are more and more funders that recommend or require the sharing of protocols and recommend protocols.io specifically. And since 2019, more and more universities and companies have been signing up for site licenses that make the private side free as well, as I mentioned a second ago. All right, so at this point, I'll exit the slides. If you're joining a little bit late, as a reminder, this is being recorded and we will be sharing the video and the slides with everyone who registered for the webinar. All right, and so going to protocols.io, like I said, every protocol that you create starts out privately, and you decide who to share it with, what permissions to give to the people you're sharing with. Can they only view? Can they view and edit? So you control that, and if and when it's appropriate, you can make it public and add it to those 14,000 public protocols that we have. So for the demo, I'll be demonstrating the functionality on one of those 14,000 public protocols, but obviously anything you create as private, it's yours, and you decide who, how, and when, who to share it with, how to share it, and when to make it public, if you decide to make it public. So the first thing to note is that we've put a lot of effort into the editor, into the visualization of the protocols. It really is a tool for capturing the details and making it easy for you to create protocols and capture the step-by-step details for you and your colleagues or someone who comes after you in 5 years to replicate and build on your results. So these are not static PDFs. You'll notice that the protocols are dynamic and interactive. Any step that you mouse over, you can ask a question on it. And you can see here on step 2, on the right corner, there are 7 back and forth questions. You can click on them and see the discussion that's been happening right directly on top of this step. So if you ask a question, it will go to the author, to everyone who's using the protocol, and you can have back and forth conversations. By default, if it's a public protocol, you will ask publicly, but you can also switch into private comment. Sometimes people add images into these questions saying, is this what I should be seeing? Is this yellow enough? Is this the kind of quality that you see in your hands? So over time, you sort of build up an FAQ directly on top of the steps of the protocol. So instead of answering a given question over and over by email, you answer it on protocols.io. You can direct people to that answer with a link. And ideally, over time, people stop asking you that question because they can see the answer right on the protocol. In terms of the dynamic and interactive aspect of it, you'll notice that this protocol has version 2. So as these discussions happen, as you see how to improve and clarify the information in your protocol, with a click of a button, you can create a new version. The older one does not disappear. We preserve everything. And you can see under the More drop down, there is actually a compare button that if you click allows you to see which parts of the protocol are the same, which ones are different. So it will be highlighted for you, and you can see steps 3 and beyond are the same. Step 2 has changes between version 1 and version 2. There are many other bells and whistles in the editor. You can add videos easily, tables with formulas, images. There are a lot of structured components that make it easy to say exactly which reagent you used. If you're spinning and centrifuging, you detail the length, the speed. So there are a lot of components that add structure to the steps and make it easy for people to, and yourself to repeat the protocols and see exactly what you're doing. And in terms of the dynamic and interactive part, every protocol serves as a template. So you can see there is the fork, copy fork button that allows you to take any public or private protocol. And, you know, if you're working with liver tissue and I'm working with kidney, there might be slight edits, slight tweaks. It's not that one protocol is right and one is wrong, but we might have slight differences between them. And so you click fork. It becomes any public protocol that you see, you can fork it with this button. And if you're signed in, a copy of it will be added into your file manager that we'll see next in a couple of minutes. And then you'll have an edit button. You'll be able to change it. You'll be able to put in those tweaks. And again, the compare functionality that you just saw in versions, that also works for the forks. So you can easily see what's the same and what's different. And if and when you're ready, you can publish a fork, and we link back and forth between the original and the fork. We try to give credit to the original authors and to the people who are sharing the tweaks as well. And under the forks tab, you can sort of see on top of the versions, what if there are any public forks. You can see what is the evolution of the protocol and what people are changing. And the last thing I wanted to highlight on the protocol level is the run button, and that's relevant for hands-on field and wet lab protocols. And of the 14,000 public ones that we have, around 5 to 10% are computational pipelines that people are reporting their workflows and pipelines on protocols.io. It's not a replacement for GitHub, but is complementary. So you put the code in GitHub, but on protocols.io, you can outline the exact pipeline, what software you've used, which data sets, so that it's clear what cutoffs, what parameters, and what was the entire pipeline, not just the code that you might have written. Inside the steps, I forgot to mention, you can attach files. So if you have scripts, Excel files, Python, MATLAB files, you can add them into an individual step. But the point is that protocols.io is used both for dry lab and wet lab methods, but for the wet lab ones, there is the run button. And that allows you sort of like a cooking checklist app. As you're performing an experiment, you can click run, and we have a mobile responsive site. So on your phone, on a tablet, on your smartphone, on a tablet, as you're doing the experiment, you can actually run through. You can see what step you're on. You can say, I'm done with number 1, on to number 2. If there is a timer that you've used, you can start it right on your iPad or Samsung tablet in the fume hood. And as you run through, you get a stamp of when you completed the step. And if you change anything, you can click edit and say, today I used 4 microliters instead of 5. And then when you're done and you save, you have a record of exactly how you ran the protocol on that particular day. You're not changing the underlying protocol, but you can have edits in the record of exactly how you ran it. Right? And then when you click save, just like the protocols, they will be organized inside your file manager. So let's switch from protocol level view into the file manager where you organize the protocols. So you're looking at a demo workspace, and the file manager is pretty much like the file manager you have for folders and subfolders and organizing your files on your own computer. This is our online file manager. You can create a structure to make sure that, whether it's by project, by species you're working with, if you have a large team and your entire group of 20 people is using it, you can organize with folders and subfolders, which projects are where, and have access to each other's protocols. One thing to note is that every workspace has a little private folder. So any protocols that you put into your private folder, even if they're in a workspace that others have access to, it's yours until you drag and drop it into the organized space for your colleagues and collaborators to see it. So anything that goes into the private folder is only visible to you. But workspaces allow you to easily organize and quickly share with a whole team. If you have a site, so we do charge per person based on the size of the workspace, not per protocol that you create. So if you have a premium workspace, it's unlimited number of protocols, but the more people are added to the workspace, the more expensive it becomes. If your organization has a site license, you can create as many workspaces as you need. So if you have an immunology component in the lab and a virology component, you can have different workspaces for different members of your company or your research lab. So you can either organize everything inside a single workspace or create multiple ones with different members, different access points, and different permissions as needed. Right. And the next thing that I wanted to highlight, you'll see in here, so if I click create new protocol, I will choose which workspace, which folder is going to go in, and it's very easy to do, and we'll create a protocol like this that you can then edit, add the title, etc. But one of the things I want to note, if you're new to protocols.io, many of you already have existing protocols, and the first challenge is not how do I publish and how do I create gorgeous, beautiful protocols using the protocols.io editor, but the first challenge is, what do I do with my existing protocols? And so first thing I wanted to say is, when you click new, you can create a new protocol, or you can just upload files. So you'll see this PDF here. The workspace, kind of like Dropbox, allows you to just take 10 existing PDFs or Word files, whatever format, or zip it all together into an archive and just put it into the workspace so that it's organized in the right place. And That takes you 10 seconds, but you already get your existing protocols into the protocols.io workspace. That's really fast, but it doesn't make your protocols dynamic or interactive. No one can fork them. No one can comment on them. You can't publish this on protocols.io. So it is limited. It's just a way to very quickly get all of your files, all of your team protocols into the same space. My recommendation, if you have a protocol, so let's create a new one as a test. My recommendation is to spend an extra 30 seconds and actually click create new protocol. Choose the template. Is it generic, biology, chemistry? So let's say it's generic. You're not committing to one path or another. A protocol can have steps that are wet lab, steps that are dry lab towards the end if you produce data that you're analyzing. So don't worry about which template you chose. And you can see in here, it says generic. So the template just defines what components I see in the editor. And I can always switch from generic to computational, and then I should see command, data set, software components, and so on. But what I was going to demonstrate is that if you have existing protocols and you have not an hour to import each one, but you have a minute for each one, what I recommend is, even if you don't have time to enter everything step by step and create a full protocol on protocols.io, if you just want to share a PDF, what I recommend is putting in a title, adding an abstract, and then on the description, so now we're inside the protocols.io editor, scrolling down and just adding your attachment to the protocol. So you can drag and drop into it. Or you can click upload. So I'm selecting a file from my local computer, adding it to the protocol. You'll see there are keywords that you can fill out to help discoverability. So instead of polymerase chain reaction, put in PCR. Instead of high performance liquid chromatography, put in HPLC so that you can easily find it in a search. I did say I highly recommend taking a couple of minutes to add the abstract. And then once you do that, if we go back to the file manager, now you can see, let's reload it. Now you'll see the new protocol that was created in the workspace. You can drag and drop it into the appropriate folder. There's the webinar test. If we go to view it, you can see the attachment. It doesn't have any steps, it doesn't have any videos or anything fancy from our editor. But the nice thing is, if you spend this extra minute per protocol, it'll be very easy to find them. It will be easy without having to download the PDF. It will be easy to see which version is this. Am I looking at the latest one? Am I looking at the one for the right strain or the right cell line? So strong recommendation to create a protocol. And if you don't have time to put in the steps, at least just use the attachment instead of simply dragging and dropping. And the nice thing is, once you've created it, even if it doesn't have steps, if you really need to, you can publish the protocol and get a DOI for it and add a link to it from a paper or share it on Twitter. And later, because we have that versioning that we're talking about, you can always come back, click new version, and make it prettier, add formatting to it, add any additional information. So you'll be able to come back, but at least you have something that you can easily share with other people and even publish it, even though it's not a full, beautiful protocol. Okay. And last thing I was going to say, if you have existing protocols, is that you can open your Word or PDF file, and instead of copying step by step, you'll see in the editor, when you go to the steps section, not description, but steps, you'll see that there is insert steps from a text file. So you can take the step-by-step instructions, just paste them in here, and they will get parsed and converted into protocols.io format in an instant. And so you can select each number as a step, each line as a step, they'll get converted, and then you can go in and edit them as needed, take advantage of the components that you see on the right-hand side. So these are some of the options. And the easiest, although not everyone has free access to it, but the easiest way to get your existing things in, especially if you are at an organization that has a site license, is to actually take advantage of the protocols.io editorial support. So if you scroll to the bottom, you'll see a link. It's in one of the slides, so you'll see it as well, you can find it on protocols.io. We enter protocols is a part of protocols.io, where if you're signed in, you can go in, drop your PDF or add a link to a Google Doc, put it in here. It will go to our editorial team. One editor will put it in, another one will check to make sure that there are no mistakes, and you'll get a notification very quickly after that when it's ready for your review. You can comment, and if it looks good, we'll reassign it privately to you for internal use or for public sharing. So again, if you already have premium through your organization, then you probably have an import code that you can enter here for free import of existing protocols. If you're at a university, you can actually go to /institutions to check protocols.io /institutions, where you can check if your university or nonprofit has a site license for protocols.io. So on that page, you see check if your organization has premium access, and you can scroll down and see if you are part of it. And if you are, then you click on it, and you'll have instructions on how to activate your premium if you haven't already done so. Right. And we're almost at the Q&A. So we'll come out of here. We've gone over protocol creation, and I think I should also mention how to share the protocols. And then publishing, we won't spend much time on because you're just getting started. We have a separate webinar on publishing, and it's also quite self-explanatory. There is a publish button there. I did not, this is a good thing to highlight, I did not specify that workspaces, just like protocols, can be public or private. So if you create a workspace, it starts out privately, but as you can see here, you can make it public. This workspace has hundreds of members. We have workspaces with 10 members, workspaces with over 500,000 members. But these public workspaces, you should check out the ones that exist in your field on protocols.io. And if none do, you can create one. But the really nice part of these community spaces is that when you share a protocol publicly, it doesn't just go into the bucket of the 14,000 public protocols, but it gets added to this community. And as you add new versions, as discussions happen, all of the members of the workspace are updated. They get notifications when there are new versions, new protocols. So it's a way to stay up to date. There is a discussion tab that allows you to ask questions and see all of the discussions, not just on the protocol, but you can just ask the people that are working in the field, that are experts in particular techniques that you're looking at. You can ask them questions. So if you go to protocols.io, you can search not just protocols, but you can search for workspaces relevant to the techniques and topics that you study. And we've gone over the file manager. I should say that we have a lot of integrations with other cloud tools and some electronic lab notebooks. So all of the files that you have in the file manager, with a click of a button, you can export all of them. You can send them to Google Drive, Open Science Framework, so Dropbox. It's very easy to do that. We'll send you all of the links. This is how to import the steps that I already showed. Protocol import service, we've gone over some tips on how to create protocols. And last thing, as I said, we won't talk about publishing in detail, but I'll just mention that if you have questions about it in the Q&A in a minute, we can go into it a little bit more. But on a protocol, you'll have options to publish or reserve DOI. So if you're an academic, we encourage you, if you're about to submit a paper, a manuscript to a journal, we encourage you to just make it public and add a link to it. You'll get a DOI and you just add a link to it. In some instances, your collaborators, your PI, or you yourself might want to keep it private until the paper is accepted. And in that case, you have the reserve DOI button that you can see here, which will give you what to put into the method section of your paper, but it won't make it fully public until the paper is accepted and you take a step of actually confirming, yes, let's make it public now. But by far the easiest is just to make your protocols public and add them to the paper. Since we launched in 2014, we've never had an instance of someone saying a journal or a publisher found it problematic that they've shared the protocol before submitting the paper. It's kind of like putting up a preprint of your paper. This is just a part of it, methods only. And we've never had an instance of someone having a problem with it. All right. And with that, I am going to stop. And as I said, our general support is info@protocols.io. My personal email is lenny@protocols.io. And a reminder for those who joined a little bit later that we do have other webinars on protocols.io/webinars. You can see what the different ones are that we offer every month. And you don't have to wait for a webinar. If you go to protocols.io/help/demo on a day and time of your choice, you can book a 30-minute session on Zoom with someone from the protocols team to ask more detailed questions. All right. I will stop there. I am going to allow, you can either put in a question if you have one into Q&A, but for those who are still here, I just permitted talking, so you don't have to type it into Q&A. We now have a small enough group. We run these pretty frequently, so if you have a question, just feel free to unmute yourself and ask. And if there are no questions, then thank you for taking the time to attend the webinar. And I'll stop recording here.